Here is the video recording of our Chapter 15, Genes and Proteins lecture. So first off, we need to define what gene expression is. So you know that your genetic material, your DNA, which is housed in the nucleus, contains genes, right? And we know that for organisms like us, we contain a set of two alleles that make up a single gene, and that those two alleles will interact with each other, and expression will be determined by both of those alleles. Even if one is dominant and one is recessive, the dominant masks the recessive, or we now know that genes have much more complex interactions than just pure dominance and recessive. In any case, gene expression is going to be a series of steps that are going to take place, which are going to take the information that's coded for in the sequence of those nucleotide bases of DNA, and that specific sequence, it gets expressed. It's going to come out in the cell's specific makeup of proteins. So genes give rise to proteins. And then these proteins will have a phenotypic effect. They will affect the individual's phenotype in some way. There are two steps to gene expression. So the first is called transcription, and the second is called translation. So in transcription, this is when DNA is going to be copied into a molecule of RNA. So we have a gene, and it's going to be read and copied, and an RNA product will be the result. Now there are three types of RNA that are made in this process of transcription. They are mRNA, that M stands for messenger, tRNA, that T stands for transfer, and rRNA, and that R stands for ribosomal. And we're going to talk about those three different types of RNA in just a few minutes. So that's the first part of gene expression, transcription, going from DNA to RNA. And the second part is going from RNA to proteins. And that second part is called translation. And so the RNA gets translated into a protein. The specific RNA that gets translated is the messenger RNA, or mRNA. That's the actual type of RNA that gets read, and based on what's there, based on that message, the protein is made. Okay? tRNA and rRNA are also involved in translation, but they aren't specifically translated themselves. Okay, so again, here are those three types of RNA, and we'll be talking about these throughout the lecture, but I want to give you a basic definition right now. So the first one, mRNA, or messenger RNA, this is going to be the RNA that gets translated. Okay, this is the copy of the gene that gets read and that gets made into, its message gets made into a protein. It is a single-stranded molecule, and again, it's going to carry genetic information that originally came from that gene. Transfer RNA, or tRNA, again, it's still single-stranded, but this is very interesting. So mRNA stays as a linear structure, where tRNA folds back on itself, and it actually forms a T-shaped molecule. Now that's not where tRNA gets its name from. It's not named because of its shape. It just happens to have that shape. Why is it named that way? Because it transfers amino acids from the cytoplasm to the site of translation. So it's going to be responsible for transferring or carrying amino acids to the site of translation, specifically to the ribosome. And we're going to talk more about the ribosome throughout the chapter. Okay? I think I said everything here. So it's involved in translation, and its specific job is to bind to and carry amino acids to the ribosome. Okay? And hopefully you remember that proteins are made up of amino acids. So the tRNA is bringing in the monomer molecules that are going to be used to build the protein according to the sequence of bases in the mRNA. The last type of RNA here, ribosomal RNA, As the name implies, this RNA is going to make up some of the structural and functional parts of the ribosome. So it's found as part of the ribosome. It's also single-stranded, but again, it doesn't stay linear like mRNA. It doesn't fold into a T like tRNA. Instead, it folds up into a globular structure. Okay, so it has a very particular three-dimensional shape, and it's going to be part of the ribosome. And it has enzymatic or catalytic functions that are going to be required during protein synthesis. It's actually going to be involved in translation. It's involved in actually catalyzing the reaction where one amino acid gets attached to the growing polypeptide chain. So it's very involved in the translation process. Okay, so that brings us to the genetic code. And so when we think about DNA, remember that DNA is composed of just 4 nucleotides, right? 4 nitrogenous bases, so A, T, C, and G. You can think of those 4 nucleotides as the 4 letters of an alphabet. They can be arranged in many different ways, many lengths, to give us the multitude of genes that we have. And so the gene, right, the message in that gene, it gets copied into an mRNA transcript, and we'll talk about how in just a few minutes. But you can think of mRNA as a mobile copy of usually just 1, sometimes more, genes. And so the mRNA, the messenger RNA, is going to be a copy of 1 gene in the DNA. And it's going to be single-stranded, and it's mobile, which means that it can leave the nucleus where the DNA cannot. So it's going to leave the nucleus through a nuclear pore. It's going to get out to that cytoplasm, and that's where it gets translated. Okay, so transcription happens in the nucleus, and translation is going to happen out in the cytoplasm. Again, the mRNA is going to be built from the DNA, not DRNA, from the DNA, and it's going to be built in a complementary fashion. So we'll talk more about the machinery and how it does that. The letters of the alphabet here for mRNA are A, U, C, and G. Remember from back to chapter 14, we learned that mRNA does not have thymine. Instead, in its place, it has uracil. Okay. Okay, so then that mRNA mobile copy goes out to the cytoplasm. It's read by a ribosome. We'll talk more about that process. And then that ribosome is going to build a protein based on the message. And we know that proteins, again, are made up of amino acids. So amino acids are going to be the monomers, right, that are going to be put together to form a polymer. And to remind you, there are 20 different naturally occurring amino acids that get put together, again, in various lengths and various combinations to form a multitude of proteins. How those amino acids are arranged, you can think of those like letters of the alphabet as well. Those are going to be arranged depending upon what's in the message in mRNA, which has been coded for by the gene. So your genes determine the amino acid sequence of your proteins. Okay? Okay. When we talk about how these amino acids, their ordering, how does your ribosomes, how does your protein manufacturing machinery know how to order them? We're going to talk about that in more detail. Again, that's called translation. But just as a kind of a precursor, each amino acid, right, the 20 different amino acids, each amino acid is coded for by a 3-nucleotide sequence in the mRNA. We call that the triplet codon. Okay, and where did that triplet codon come from? It came from the DNA. So your gene, again, gets transcribed into RNA. And the RNA, we say, gets read 3 bases at a time. That's called a codon. And as the ribosome reads those 3 bases, it's going to bring in a very specific amino acid. So each codon, each triplet of bases in the mRNA is going to code for a specific amino acid. Okay, so let's think about that. There's 20 different amino acids. And those 20 different amino acids you learned last semester, they have the same backbone. Remember, there was a central asymmetric carbon bonded to an amino group, bonded to a carboxyl group, bonded to a hydrogen, and then something else. So the only difference between the 20 amino acids is what's here, the side chain, which is represented in all of these as this blue part. And you can see that we can group the amino acids based on this side chain. So for example, side chains that don't have any really polar atoms or highly electronegative atoms, they're going to be considered non-polar, right? When we do have charge, right? So we have a positive charge there. We've got this highly electronegative oxygen here and here. These guys are going to be all polar. We go here. These are nonpolar containing rings. These are negatively charged. So again, polar side chains. Positively charged. Again, polar side chains. And so the gene in the DNA dictates the code of the RNA, mRNA. And the mRNA is going to be read 3 bases at a time, and that's going to tell us the sequence of the amino acids. And the sequence of the amino acids is going to tell us, based on these properties that we see here, how that protein folds, right? And so remember the lock and key? A protein's final 3-dimensional shape, how it finally folds up, that's all based on the message in the mRNA, which is based on the DNA. And that protein, once it folds into its 3-dimensional shape, then it's functional. If there is a mistake, if there's a mutation in the DNA, and that gets carried to the RNA, that can sometimes be carried into protein structure. And if a protein doesn't fold properly, it probably won't work properly. Okay, so this whole process of transcription and translation, what it ends up giving us is a sequence of amino acids that's very, very specific to the protein's final shape, and therefore it determines whether that protein will function or not. So it's very, very important. Okay, so here's this thing called central dogma. And I introduced this in lab before break. I'm not sure if you guys remember that or not. Central dogma pretty much says what I've been saying all along. So we have genes, right? We have our DNA, which is genes. Those will specify the sequence of bases in the mRNA product. That's being shown up here. So DNA is shown in blue. The DNA gets opened, it gets read by RNA polymerase. RNA polymerase lays down bases in the 3' to 5' direction. Eventually, this transcript of RNA comes off. The DNA will reseal up. Okay, and then if we're looking at eukaryotes, organisms like us, there's some processing that happens to that RNA. Then that RNA goes out to the cytoplasm. And in the cytoplasm, it gets read 3 bases at a time, or 3 codons. Or sorry, 1 codon, 3 bases at a time. And as it gets read, here's the amino acids. They're going to get added according to the message. So the mRNA is shown here in this, I don't know, orangey, peachish color. Okay. And those amino acids get put together, and finally we end up with a protein, and that protein folds up and it does its function. So again, that's central dogma. So when you hear central dogma, I want you to think DNA to mRNA to amino acid sequence in the protein. Okay? And there's a whole lot on this slide that we haven't covered yet, and we will. I just want to let you know, we haven't talked about tRNA or rRNA very much yet. We will. I do need you to know that even though mRNA is the type of RNA that gets read or translated, rRNA and tRNA are really important in the decoding, in translating that message. They have a very specific role. So what does the rRNA do? Well, it's part of the ribosome, and the ribosome is required to read the mRNA transcript, and it's required to build the polypeptide. And where does the tRNA come into play? Well, you can see this right here bringing in arginine, and there's this GCU here at the bottom. This is the tRNA molecule. This is called the anticodon. We'll talk about that in more detail in just a minute. And it's bringing in an amino acid. And these two amino acids here, phenylalanine and methionine, these were all brought in by tRNA molecules. So again, T stands for transfer. What are they transferring? They're transferring amino acids from the cytoplasm to the site of protein synthesis, so to the ribosome. Remember, the DNA here, it's just copied, right? But it stays protected and intact in the nucleus if it's a eukaryotic cell. If it's a prokaryotic cell, still it stays intact in a region called the nucleoid, not surrounded by a nuclear membrane, but still in a specific region. And so RNA is kind of like the middleman, right? And RNA is the copy of your genetic information, and it has specific jobs. If it's mRNA, it gets translated. If it's rRNA, it's part of the ribosome. If it's tRNA, it's going to be involved in transferring amino acids into that site of protein synthesis. All three molecules of RNA are very, very important. And in this process of translation couldn't happen if any of them were missing. Okay, here's another figure just showing central dogma again. So here's our double strand of DNA. You can see it's helical. We've got our nucleotide bases here in the center. We've got our sugar phosphate backbone on each side. The two strands are running anti-parallel to each other. Okay, all of that should be familiar to you because you've finished chapter 14 by now. And so what's going to happen is where the gene is, right, the DNA will be unwound and unzipped. And one strand of the DNA will serve as the template. It's the one that gets read, while the other strand here is the non-template strand. We'll talk about those in more detail a little bit later too. And so the template strand is going to be read, and the transcript shown here in pink, this is the RNA transcript, will be produced. Okay, that will then be read 3 bases at a time, or codon at a time, by the ribosome. And the tRNA molecule will bring in the appropriate the appropriate amino acid according to the codon. And so AUG, that actually codes for start. And when we start, we always bring in the same amino acid. It's methionine, or MET. Codon number 2 here reads ACU. That will cause the tRNA molecule carrying threonine to be brought into the ribosome. So that'll be our second one. And then this third codon, UGC, codes for cysteine. That'll be brought in. Codon number 4, GAA, codes for glutamine, and so on and so forth. Okay, so going from DNA to RNA to protein. That's central dogma. Okay, now I don't expect you to memorize the genetic code, but I do want to take a peek at it, and I want you to know that the genetic code is universal. What does that mean? That means that, for example, UUU, right, that codon of uracil, uracil, uracil in the mRNA transcript is always going to cause the tRNA carrying phenylalanine to be brought into the ribosome. Okay? It doesn't matter if we're looking at humans or if we're looking at gene expression in bacteria. UUU always codes for phenylalanine. So that's when we say the genetic code is universal, that means that it doesn't matter what organism we're looking at, these codons are going to be read the same. They code for the same amino acids. Here's some evidence for that. So this is a plant, okay? Now this plant doesn't normally bioluminesce. However, if I take the gene from, let's say, the jellyfish, just like we were going to do in lab, and I put it into this plant, right? So I just genetically modified the plant to carry and express the bioluminescence gene. Well, even though the plant and the jellyfish are 2 different organisms, they're both eukaryotes, but they're pretty distantly related to each other. Even so, the machinery responsible for gene expression in the plant can still read the gene that codes for bioluminescence, even though it's from a different organism. Here's another example. So humans, right? We have a gene that codes for insulin. Insulin is required. It's a required hormone in our body, and without it, there's very detrimental effects. So people, for example, with diabetes who are lacking insulin or lacking the ability to produce insulin, they need to take insulin. Where does that insulin come from? Well, we've genetically engineered bacteria in the lab. We've put in the human insulin gene. It's been modified a little bit because there is some differences between prokaryotes and eukaryotes. But for the most part, the gene is the same. And so the mRNA transcript, it's definitely the same. And the mRNA transcript inside of bacteria, it gets read the same exact way. The codons are the same, and those codons code for the same amino acids. And so the insulin protein can be made, the human insulin protein, can be made in bacterial cells. Again, why? Because the genetic code is universal. These codons here in this table, they are going to code for the same amino acid, whether we're in bacteria or plants or jellyfish or humans. So the genetic code, again, is universal. It's also redundant. So what does that mean? Well, if we think about it, okay, we have 20 different amino acids. But if you look at this table, there's 64 codons. So there's 64 three-letter combinations that there can be, but there's only 20 amino acids. So let's take a look here. So what does it mean to be redundant? Let's look here at phenylalanine. So both UUU and the codon UUC, both of these will trigger tRNA containing phenylalanine to come to the ribosome. So both of those codons code for the same amino acid. The same thing here, UUA, UUG, redundant. Both of them will cause the tRNA molecule containing leucine to come in to the ribosome. Same thing here, all of these code for leucine. CUU, CUC, CUA, CUG, okay? Some that are not redundant. So here, AUG, which is also called the start codon, this codes for methionine. Nothing else on here is going to code for methionine. I also want to point out the stop codons. Okay, UAA, UAG, and UGA. When the ribosome gets to any of those three codons, it's going to cause the ribosome to stop. Okay, so I need you to know that the genetic code is coded for in the bases of the mRNA message, which is coded for by your genes in your DNA, and that the genetic code is universal and that it is redundant. Okay, so I'd like you to pause here. This is our first Are You With Me? Take some time and answer these questions right in the Are You With Me sheet that, again, you're going to upload right to Blackboard when you're finished. So before we go on, I got a couple questions for you. So remember, these videos are kind of cleaved together. So some of this I recorded earlier, and some of it I'm adding to it right now. So here's some added parts. So a couple questions for you before we move on to the second part of this chapter. So here's the first one. The AUC and AUA, so adenine, uracil, cytosine, and adenine, uracil, adenine, both of those codons, so those are triplets of bases found in mRNA, so we call those codons. Both of them specify the amino acid isoleucine, okay? What feature of the genetic code explains this phenomenon? So is it red or pink? It's supposed to be pink. I think it's reddish. Is it DNA is complementary? Is it blue? There are nonsense codons. Is it green? The genetic code is universal. Or is it yellow? The genetic code is redundant or degenerate. So take a minute to think about that. Pause me if you need to. So again, the fact that AUC and AUA, both code for isoleucine, tells us that the genetic code is redundant or degenerate, right? That multiple codons code for the same amino acid. Now, if you answered the genetic code is universal,
yes, the genetic code is universal, but this example here, they don't relate. Okay? So the answer would be yellow. Next question. How many nucleotides are in 12 mRNA codons? Okay? So you gotta do the math. Give you a minute. Okay, so every codon has 3 bases, right? Because the mRNA, the genetic code, the codons there, that's a triplet. So 3 bases. And so if there's 3 bases to 1 codon, if there's 12 codons, that's 12 times 3, so that's going to be 36. So there's 36 mRNA nucleotide bases in 12 codons. Okay, now for this one, you're going to have to flip back to the genetic code figure, the box where it shows you if this base is followed by this base, this is the amino acid that you would get. So you got to kind of flip back to that in your notes or in your textbook. So here we go. It says if 6 bases within a gene are cytosine, adenine, thymine, cytosine, guanine, threonine. Or sorry, thymine. Getting my amino acids and my bases mixed up here. So CAT, CGT, if that's what the DNA sequence reads, what will the mRNA transcript read? Okay. And that gene, that's the template or sense strand that we're talking about. Okay, so what will the mRNA transcript read? What will be the sequence of the bases in the mRNA? And what amino acid sequence will there be? Okay, so the easy part is figuring out the mRNA sequence. Okay, you just do simple base pairing rules. The hard part, which you might have to refer to your textbook, I have mine right next to me, is for the amino acid sequence. So let me give you a few minutes to do that. Again, pause me if you need a little bit longer. Okay, so the the mRNA sequence of bases is going to be G, U, A, G, C, A. Okay, so for both the pink and the blue, it seems right. So now you've got to go over to your genetic code box in your textbook or in your notes. And the first letter of our mRNA code here is G. So you got to find the G. Second letter, which is across the top, is U. And our third letter is A. And so G, U, A, that would be the codon. That codes for Valley, so VAL. Okay, so that'd be our first amino acid. The second amino acid is based on the next 3. So that's GCA. So GCA codes for alanine. Okay, so the answer here is blue. Okay, next question. Knowing that there are 20 different amino acids and 64 potential codons, how many potential proteins are there? So I'll give you a second to think about this one. So 20 times 10 to the 64th, 64 times 10 to the 20th. Is there an infinite number, or is it 2 times 106 to the 4th? So yeah, there's an infinite number, right? Because proteins can be various lengths, various sizes in the end. And there could be tons of different, orders of those amino acids, and there's 20 different of those 20 different amino acids that could be ordered in a bunch of different ways. And so, and because the lengths vary, right, there really could be an infinite number of proteins. And we definitely haven't identified all the proteins yet. Okay, so that's it for this part. So I'm going to let you go ahead and go on to the next part, and then I'll be back on for some more questions with you.
Okay, so we've got kind of the basics. Now we're going to get into kind of the nitty-gritty. And we're going to start off with prokaryotic transcription. And so studying transcription, right, for this first part of gene expression, then we'll do translation. Studying transcription in prokaryotes has proven to be easier than studying transcription in eukaryotes. And so a lot of what we know about transcription comes from those studies. We will talk briefly about transcription in eukaryotes as well. There's really not too much of a difference, but there are some things that are different. Okay, so again, when you hear the term transcription, you're thinking this is the initial step in gene expression. This is where we go from our DNA in the here it's in the cytoplasm. We go from our DNA to an mRNA transcript. And remember, this is the same thing. This could be tRNA or rRNA. Okay. Again, there is no nucleus in these prokaryotic organisms. And so even though we're going to talk about transcription and translation separately, keep in the back of your mind that they actually take place simultaneously. Because there isn't a membrane separating the nucleoplasm from the cytoplasm, both transcription and translation can happen at the same time. We'll come back to that after. So the first step in transcription is going to be the unwinding and unzipping of the DNA right at the gene. Okay? And what do we form here? We form a transcription bubble. If you remember back to chapter 14, when we were talking about DNA replication, we said the DNA was unwound and unzipped, and we formed a replication bubble. It's the same idea here, except for now you have a transcription bubble. Okay? And only one strand of the DNA is double-stranded, only one strand actually contains the gene. And so only one strand will be transcribed into RNA. We call this strand, we can either say the template strand, or you would call it the sense strand. The other strand that does not contain the gene, this particular gene, that other strand is going to be called the non-template or the antisense strand. Okay? So the DNA gets unwound and unzipped, and then we start. There's 3 steps in transcription, and they're named really easily. They're called initiation, which is followed by elongation, which is followed by termination. And so this first part, initiation, is going to happen at a region called the initiation site. Now, before I get into that any further, I want to just back up for a second. And we talked about how when the DNA gets unwound and unzipped and we have this transcription bubble, that one strand of DNA is the template and the other one is the non-template. Okay, now that's going to be specific for the gene. So what do I mean by that? Well, let's look here at gene 1. Notice how the 2 strands of DNA are colored differently. So we have one strand that is dark blue and one strand that is light blue. And so for gene 1, notice, here's my RNA polymerase. This is going to be the enzyme that's going to actually be doing the copying of the DNA, or the transcribing of the DNA into RNA. It is going to be reading the light blue strand here. And so in pink, we have our transcript coming off. And so for gene 1, the light blue strand is the sense strand, or the template strand. This is the strand that actually contains the gene. You might say, well, what's the sense of the antisense, right? What's the sense of this non-template? Why is it there? Well, if we go downstream and we look at this gene, gene number 2, it's actually the opposite. The dark blue strand of DNA holds the gene in this case. And if we go down to gene 3, we just reversed it again. In gene 3, the light blue strand contains the gene. And so it's not true that one strand is always going to be the strand that contains the gene. It's going to depend on the gene. Okay. So let's get into initiation. So initiation, there's going to be a region upstream, okay, before preceding the actual gene, okay, in the DNA. It's going to be called the promoter sequence. This is a sequence that's going to come before the gene, so we say it's upstream of the coding sequence. And it's going to have a very specific nucleotide sequence. Again, this is in the DNA, which is going to draw the RNA polymerase. This is the enzyme that's going to be doing the synthesizing of RNA. It draws that RNA polymerase to that site. Okay? Now, there are some consensus sequences that we find that are similar in all promoters. Okay, and so you can look here. This represents the double strand of DNA, what I'm dragging my line across here. Okay, the gene is going to be down here in green. Before the gene, so upstream of it, we've got this region called the promoter. Okay, and here at the -35 spot, there's always this sequence, TTG, ACG. And at the -10 spot, so 10 bases upstream from our start site, there's going to be something called the TATA box, which is TAT, AAT. These are consensus sequences. They're similar in all promoters. These are going to be the regions, right, that are going to draw the RNA polymerase to this site. And so here in orange or peach, we've got our enzyme. That's our RNA polymerase. And so initiation is going to be when the RNA polymerase binds to the promoter sequence. Now, there are other proteins that are involved in this binding event. So, for example, this blue protein here is called sigma factor. This is part of what we call the holoenzyme. And there's actually more proteins than just the sigma factor. They're all required to correctly recognize and for the RNA polymerase to actually bind here. So it's not just RNA polymerase that binds, it's a whole bunch of other factors. Okay, in particular, sigma factor for prokaryotes. Once the RNA polymerase binds there, right to the correct promoter sequence, it's going to start to unwind and unzip the DNA, and then transcription will start. Okay, so again, initiation is pretty much RNA polymerase binding upstream of the gene at the promoter sequence. What comes next? Elongation. And so at this point, any other proteins that were helping, so for example, the sigma factor that we saw in that previous slide, they're going to be released from RNA polymerase. And so now RNA polymerase is going to pretty much do this part on its own. So RNA polymerase will read the template strand. So if you look in this figure here, RNA polymerase is shown in orange. It's unwound and unzipped our DNA. We have our template strand, which is the one that's going to be read. And we have our non-template strand, which is not going to be transcribed. And so RNA polymerase is going to read and it's going to lay down bases in the 5' to 3' direction, always adding to the 3' end of the growing strand. And it's going to continue laying down bases that are going to be complementary to the template strand. So notice when RNA polymerase sees an A, it puts down a U. When it sees a G, it puts down a C. When it sees a T, it puts down an A, and so on and so forth. So the RNA bases are added in a complementary fashion to what is stated, right, the sequence in the template strand. And as the RNA polymerase is made, it's going to come off of the DNA. And so these bases down here that have already been copied, these bases down here, that have already been transcribed, they're going to re-anneal. So hydrogen bonding will reform. So the RNA transcript kind of hangs off of the DNA as it's being produced. Okay. And so elongation continues until we get towards the end of the gene. And towards the end of the gene, we're going to have termination. And termination looks a little different. Termination can either be what we call row-dependent, rows of protein that we'll talk about, or row-independent. And actually in eukaryotes, there's a whole bunch of different ways that this can happen. So let's talk about these two possibilities in prokaryotes. So row-dependent termination. This is when this protein called Rho, RHO, it's actually going to kind of truck along, follow along behind RNA polymerase. And as RNA polymerase nears the end of the gene, it will encounter a stretch of repetitive G nucleotides. So guanine, 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 guanine. And as it encounters those, it's going to stall. It kind of slows down and it stalls out. And what happens is this Rho protein that was following behind it, it doesn't slow down, so instead it kind of just jams right into the RNA polymerase, literally knocking it off of the DNA. So that's one possibility. And once that RNA polymerase is kind of knocked off a little bit, the whole complex will come apart and the mRNA transcript or tRNA or RNA transcript will get released. Okay, so that's called row-dependent termination. Again, at the end of the gene, there's a repetitive sequence of gene nucleotides. RNA polymerase stalls and it gets bumped off by the row protein. The second type of termination is called row-independent termination, and the row protein is not involved here. Instead, it's just the sequence of bases towards the end of the gene. So as we near the end of the gene, there sometimes is a cytosine guanine-rich region, and as the RNA polymerase starts to transcribe that into the mRNA, that causes the mRNA to start to fold back on itself, forming a structure called the hairpin structure. And then RNA polymerase, as it starts to reach an AT-rich region, okay, so another repetitive region rich in adenine and thymine, it will transcribe A and U, right, as complementary bases to that. And that AU is going to be very, very weak, weak interaction. And so therefore the mRNA starts to already kind of be lifted off. And then you've got that hairpin structure that's already formed, which creates even more tension. And so both of those together causes instability of the complex. And therefore the RNA polymerase will disassociate from the DNA and the mRNA transcript gets released. Okay, so in both events, as we near the end of the gene, we do have some sequences there at the end of the gene that are going to cause instability of the complex. So that's termination. Okay, so here it is kind of in a nutshell, and I'm missing the top of this. What this says up here is RNA polymerase binds to the promoter region in the DNA. So this first part here is showing initiation. Okay, so here's my double strand of DNA. In yellow, I have my RNA polymerase, and this red region right here is the promoter. So the promoter is going to get unwound and unzipped, and then as soon as we hit the first part of the gene, we will start to transcribe it. So the promoter itself does not get transcribed. This is initiation. We come down here. This is elongation. So the RNA polymerase is going to just move down the DNA double helix, only transcribing the template strand, which is shown here by the blue strand, the dark blue strand. And as the RNA transcript comes off, the DNA that's already been transcribed will re-seal up or re-anneal. And this process continues, right? Unwinding of the DNA, unzipping of it, and then rewinding and rezipping of it, until it reaches some sequence here at the end, whether it's a row-dependent or row-independent. There's going to be a sequence here called the termination sequence, which again is usually a highly repetitive sequence. And once that RNA polymerase gets here, that's going to cause instability of the complex, causing the RNA polymerase to come off, causing the RNA transcript to be released, and our DNA goes right back to normal. It's going to re-anneal and rewind right up. So that's it. We've got initiation, elongation, and termination. Those are the three stages. Now, I told you I would mention eukaryotes. So eukaryotic transcription is fundamentally the same process as what we see in prokaryotes, pretty much the same steps. So still initiation, elongation, and termination. Here's a couple of differences. So the first thing is that transcription and translation can't happen at the same time. So transcription is going to happen inside of the nucleus, and the mRNA transcript, once it gets produced, it has to be exported out of the nucleus before it can get translated. And we'll talk about that in a second. The second thing is RNA polymerase in prokaryotes. It's pretty simple. You've got your RNA polymerase, you've got sigma factor, that's going to comprise kind of the holoenzyme. But in prokaryotes, it's much more complex. There's 3 different kinds of RNA polymerase that will produce the 3 different kinds of RNA molecules that we have. So there's a specific RNA polymerase that makes mRNA, a specific RNA polymerase that makes tRNA, and a specific RNA polymerase that makes rRNA. What else? Well, when we look at initiation in eukaryotes, there's still this promoter sequence that's upstream of the actual gene, and we still have that TATA box and some other consensus sequences as well. But in this case, the holoenzyme is actually going to be built of a lot of different proteins. So you can see here TF2D, this is transcription factor 2D, transcription binding protein. There's a whole bunch of different proteins that will bind to the promoter region and attract RNA polymerase to that region. So it's just much more complex. There's more players in the game. Okay. Elongation, we still have RNA polymerase, which is going to just kind of move along the DNA. If we're making mRNA, then it's going to be RNA polymerase 2. It's still going to work in the 5' to 3' direction. So again, pretty much the same. Here's one key difference. In prokaryotes, there are no histone proteins organizing the prokaryotic genome, but in eukaryotes there are. And so in organisms like us, we've got 46 chromosomes, and those chromosomes are linear, and they are going to be organized by histone proteins. These are proteins that are going to have the DNA kind of coiled around them, organizing, making sure the DNA isn't damaged in any way. Those aren't there in prokaryotes. And so in eukaryotes, we have histone proteins that have to be moved whenever a gene is going to be transcribed. Okay, last step, termination. It's different depending on the RNA polymerase that is being used, but here's something that is the same. Again, there's usually some repetitive sequence that we call the termination sequence, and that's going to create, again, instability in the complex, and it's going to cause all of the component parts to be released and the transcript to come off. Okay, so before I go any further in chapter 15, I'm going to have you pause the video here, and you're going to answer these couple of questions. When you're finished, you can go on.
Okay, so before we go on to this next part, I got some more questions for you. Okay, make sure that you're all following along. So the promoter region, what is it? Does it contain consensus sequences? Is it found downstream of the gene? Is it involved in termination? Or is it where ribosomes bind? I'll give you a second to think about that. And so hopefully you answered the promoter region contains consensus sequences. Okay, so before we talk about that, the next one down says it's found downstream of a gene. It's not, it's found upstream, which means that it's found before the gene, okay, before that +1 site, which is where transcription starts. So it's found before then. It's definitely not involved in termination, and we're talking about transcription, not translation, so ribosomes don't even come into play there. So let's talk about these consensus sequences. These are sequences that are similar amongst all promoters, and especially in prokaryotes, okay, and in eukaryotes as well, but the consensus sequences are a little bit different if we compare the two. What is the same is that both prokaryotes and eukaryotes, they both contain the TATA box, remember that sequence of thymines and adenines at that -10 spot, so 10 bases before that initiation, that start site, that +1 site. In prokaryotes, remember there's also that -35 site where there's another sequence there. Remember, consensus sequences, again, are going to be similar between different organisms. So that's where we find that promoter region. And what is that promoter region? Remember, that's going to be the spot that's going to draw RNA polymerase and any associated other factors with it right to that spot. Okay? And there's going to be different sequences in the promoter region which are going to tell us how often that gene is transcribed. If you have a chance, we're not going to get to it, but if you have a chance to read chapter 16 about gene regulation, there's a lot more information in there about that, which is pretty cool and interesting. Again, unfortunately, we don't have time in the course, but go ahead and take a look at that if you're interested. So that's the promoter. Second question, transcription ends. Okay, so when does it end? Does it end upstream of the promoter sequence? So upstream means it comes before that. Does transcription end during elongation? Does transcription end once RNA polymerase reaches a termination signal? Or does transcription end when the RNA polymerase reaches the end of the chromosome? I'll give you a minute to think about that one. Okay, so remember that termination is going to end green when the RNA polymerase reaches the termination signal. And that's how exactly that happens is going to look different depending on the gene. Remember we talked about row-dependent and then row-independent termination. And then if we get into eukaryotes, it's even more complicated. But what is the same is RNA polymerase reaches some type of a termination signal. And that's going to trigger RNA polymerase to stop transcribing and for the complex to come apart. A couple more questions for you. What feature of promoters can be found in both prokaryotes and eukaryotes? And if you were paying attention, I just answered this. Is it red or pink? The GC box, blue, the TATA box, green, the -10 and -35 consensus sequences, or yellow, the start codon? I'll give you a minute. Okay, so the TATA box, that's going to be similar. Its location is a little different. In prokaryotes, it's at -10, and in eukaryotes, it's a little bit farther upstream. It's usually at about -30. But in either case, it's there. So the TATA box is going to be a common feature in promoters for both prokaryotes and eukaryotes. That's it. Okay, next question. Eukaryotic and prokaryotic transcription differ in that eukaryotic transcription, is it more complex and can occur while DNA replication is proceeding? Does it occur simultaneously with translation? Does it occur in the nucleus? It involves more enzymes. Or yellow, it takes longer and usually results in more mistakes. So I'll give you a second there. So hopefully you answered green. So remember, in eukaryotes, we have that nuclear membrane, and the DNA is housed inside the nucleus. In prokaryotes, that's not true. So in prokaryotes, transcription happens in the cytoplasm, and it can happen simultaneously with translation. Okay, in eukaryotes, it happens in the nucleus, and there's just a whole heck of a lot more enzymes involved in the process. Okay. Transcription and DNA replication, if you looked at the pink answer, those are not going to happen simultaneously. During DNA replication, remember that the DNA is going to be tightly coiled up into chromosomes, not accessible to the transcription machinery. And so transcription and replication are not going to happen together. Okay, so again, the answer here is green. Eukaryotic transcription occurs in the nucleus, and it's going to involve more enzymes. Okay, so that's it for these questions. So you're going to go ahead and you're going to start the next section, and then we'll have some more questions after the next Are You With Me?
Okay, so we've finished transcription in prokaryotes, and we've almost finished transcription in eukaryotes. And so after the mRNA transcript is produced in eukaryotes, it has to undergo some processing before it can go out to the cytoplasm where it's translated. This is not true in prokaryotes. So in prokaryotes, the mRNA that is made gets translated right there and then. But for eukaryotes, we actually call it pre-mRNA. And that, again, has to undergo some processing before it can be released out to the cytoplasm and be read to make proteins from. We're going to go through this with mRNA, but the same thing is true for tRNA and rRNA. Both of those have to also go through some processing before they go out to the cytoplasm to do their jobs as well. So let's go over some of these processing events. So mRNA gets made in the nucleus, and before it can leave, there's a couple of things that have to happen. The first thing, at the 5' end of the mRNA transcript, we put on a cap. It's called the 5' cap. What is it? It is a chemical modification. And so this molecule called 7-methylguanosine, it's a cap, it gets added to that 5' end of the mRNA transcript. Why does it get put there? Well, we think the reason for this 5' cap is to decrease the degradation, the breakdown of the mRNA transcript so that it can last a little bit longer once it's out there in the cytoplasm. Remember, there's lots of enzymes in the cytoplasm that could easily break down this mRNA transcript. And so to prevent it from getting kind of chewed up by the cell's machinery, this 5' cap gets added. Okay, why else? We think that the 5' cap allows the ribosome to recognize the transcript and therefore bind to it. Okay, so it also adds in recognition by that translation machinery, the ribosome. So that's at the 5' end. At the 3' end of the mRNA transcript, there is added what we call a poly A tail. Okay, there's an enzyme that is going to add a whole bunch of adenine nucleotides to the 3' end of the transcript. That enzyme is called the poly A polymerase, and it's just going to throw a bunch of adenines onto the 3' end, upwards of 200. Why does that happen? Again, we think it helps to aid in inhibiting degradation of the mRNA molecule, and it also helps the mRNA molecule get out of the nucleus. So remember, anything going in or out of the nucleus has to go through a nuclear pore, and it's relatively difficult to get in or out of that structure. And so this poly A tail, we believe, allows it to get through there with a little bit more ease. What else? Well, in the eukaryotic genome, there are regions, sometimes within a gene, a lot of times within a gene, that aren't really part of the gene. They don't really code for anything. In fact, if they stayed in the transcript, they would actually screw up the amino acid sequence and we wouldn't get the right protein. Okay, so there are two parts of eukaryotic genes, and therefore two parts of what we see in our pre-mRNA transcript. We have regions called exons and regions called introns. Exons are the important part. They're the coding regions. We want to keep those. But introns, we don't want those in our transcript. Why are they there in the first place? We're not entirely sure. They might be involved in gene regulation, turning genes on, turning genes off, but they don't actually code for the protein products. And so they need to be cut out. Okay? And the exons, the actual coding regions, after these introns get out, they have to be spliced together. Sometimes there are enzymes involved in this process. They're called SNRPs, SNRNPs. And then sometimes they're not involved. The RNA actually might facilitate this on its own. In this particular figure, we're showing SNRPs being involved. And so this whole structure here is called the spliceosome. We've got our RNA transcript here. This whole line is the RNA transcript. And we've got region 1 here, which is an exon. And over here we have exon number 2. Those are the coding regions. And then all this green that's being wound here, that is all part of an intron. And so it needs to be removed. And so these are those SNRPs, SNRNPs. These are going to be, again, those proteins that are going to catalyze this. They're going to take that intron and literally cut it out, and then they're going to splice or put together exon 1 and exon 2. So this is the intron, all the non-coding, we're not sure why it's there. And then here are the 2 exons that have gotten spliced together. Okay, here's another example. This is showing the 5' end and the 3' end, and exon 1 intron, exon 2 intron, exon 3. So this is a primary RNA transcript or a pre-mRNA transcript. The processing that would happen at the 5' end, we get that cap. At the 3' end, we get that poly A tail. We remove the introns here in green, and we splice together exon 1, 2, and 3. So that's RNA processing. Again, this is only going to happen in eukaryotes. Prokaryotes do not get any processing done. They do not have introns. They do not require the 5' cap or the poly A tail. Okay, so take a break here and go ahead and answer these Are You With Me's. When you're finished, you can go on.
Before you go on, let's just answer a couple questions together. Maybe. Here we go. Okay, so just 2 questions for you. During mRNA processing, what happens? So pink exons are removed and introns are spliced together. A 5' cap is added. A 5' poly A tail is added. Or yellow, mRNA is exported to the cytoplasm. So which one of those is correct? So hopefully you answered blue. Okay, so let's look at the pink one first. It says exons are removed and introns are spliced together. It's the opposite. Introns are cut out and exons are spliced together. It's the exons that contain the coding region. Green, there is a poly A tail added, but it's to the 3' end. Okay, the opposite side of the mRNA transcript. And yellow, mRNA is not exported until after RNA processing occurs. Okay, so blue, that 5' 7-methylguanosine cap is added to that 5' end. And the next question is actually about that cap. So what is that 5' cap there for? Does it aid in export from the nucleus? Does it prevent degradation, the breakdown of that transcript, and assist in translation once it's out there in the cytoplasm? Does it speed up the splicing events? Or does that 5' cap replace an intron? So let me let you think about that for a minute. And hopefully you answered blue. Okay. You might be wanting to answer the pink, aids in export from the nucleus. That's the 3' poly A tail. The 5' cap is there to prevent it from being broken down, and the second part of that, it assists in translation. So it actually helps the ribosome in binding to the mRNA transcript once it's out in the cytoplasm, which we're going to talk about pretty soon. Okay, so we're finished with these questions. Go ahead and we're going to go to the next part. And I'll be back on in a little bit to ask you some more questions.
Okay, so we've finished transcription. So again, we went from DNA to RNA. And if we're talking about eukaryotes, that transcript then gets processed before it can be read out in the cytoplasm. Okay, so now we're going to get into translation. So we finished transcription, we're moving on to translation. So again, genes are ultimately translated into proteins. Proteins are going to be the functional components of your cells. They are what is coded for by your genetic material. Again, a reminder, what are they made of? They are macromolecules made up of the monomeric subunits called amino acids. There are 20 different naturally occurring amino acids. And through this translation event, what's going to happen is amino acids are going to be covalently bound together according to the message in the mRNA. Okay, there's that peptide bond that's formed between these 2 amino acids. Proteins can vary in length. They can be anywhere from 50 to over 1,000 amino acids in length. Again, we have a 20-letter alphabet that's going to determine how the amino acids are going to be put together. All of that is going to be determined by the mRNA transcript. Okay, so this process of protein synthesis, this process of translation, in reality, it's going to require over 100 kinds of macromolecules. It's going to require a whole bunch of different proteins. It's going to require rRNA and components of ribosomes. It's going to involve mRNA and amino acids that'll be linked to tRNAs. I'm going to give you kind of the simplified version, but again, I want you to remember that there's over 100 kinds of different molecules that are actually involved here. Okay. I want to remind you of that genetic code. So what translation is it's when we take the information that was transcribed in the mRNA from the DNA, and we're going to use that information to build a polypeptide. That mRNA is going to be read by the ribosome 3 bases at a time. So when we talk about the genetic code, these are codons, right? So 3 bases. This is mRNA. So these are the 3 bases in the mRNA and what they code for, which amino acid they code for. Okay, so when you hear codon, this is 3 amino acids in the mRNA. The mRNA gets read 3 bases at a time, a codon at a time. And each codon specifies an amino acid. Remember the redundancy of the genetic code, okay? And remember that there are stop and start codons. So translation always starts on the mRNA molecule with the codon AUG. That's always going to be at the very beginning of every mRNA transcript where start is supposed to happen. That's going to bring in the first amino acid, which is always methionine. And then as we go towards the end of the gene, towards the end of the mRNA transcript where the gene is going to end, we're going to find one of these 3 stop codons. So UAA, UAG, and UGA all code for stop. Okay. So I told you that there's a codon in the mRNA, right? And that each codon codes for a specific amino acid. And then I told you that the job of tRNA is to transfer amino acids from the cytoplasm to the site of protein synthesis, the ribosome. And so let's talk about tRNA now. So when I say that they bring amino acids in, they're actually going to be covalently linked to the appropriate amino acid. And there's a certain enzyme that is going to covalently link amino acids to tRNA molecules. And that enzyme is called the aminoacyl tRNA synthetase. And when a tRNA molecule is carrying amino acid, it's called an aminoacyl tRNA. The tRNA molecule gets attached to the amino acid. This is the enzyme that catalyzes this. How does this enzyme know which amino acid to put onto the tRNA molecule? The tRNA molecule is going to have an anticodon. Remember, we heard codon in the mRNA. The anticodon will be complementary to the mRNA's codon. And so this anticodon, it will be recognized by the enzyme, the aminoacyl tRNA synthetase. And that enzyme is going to covalently link the appropriate amino acid based on the anticodon. So it's all very, very specific. Let me show you that. These are just 2 different pictures of tRNA. I want to make sure you can see the bottom. This is the simplified figure that comes from your book. This is a little bit more complex of a figure. It really shows the different bases. So here's the 5' end of this tRNA transcript, and if we follow this loop, look, it looks like a T, right? All the way down here, all the way up this side. This would be the 3' end. So it was linear, but because of some hydrogen bonding between the bases, it ends up forming kind of a T-shaped structure. And at the very bottom of that T, down here in loop 2 we call it, there is 3 bases which constitute the anticodon. Okay, now in this more simplified version over here, the same thing. So down here at this bottom, which would be loop 2, there would be a triplet anticodon, AAG. The aminoacyl tRNA synthase, that enzyme, will look at this sequence and it will attach the appropriate amino acid based on this sequence. Now this anticodon will be complementary to the mRNA transcript. So the mRNA transcript where this will bind to would read UUC. Okay, and UUC, if you look back at that table, codes for phenylalanine. Let's actually do that. So again, UUC in the mRNA codes for phenylalanine. And here's my tRNA, AAG, which is complementary to UUC. It's going to have attached to its 3' end the amino acid phenylalanine. And so the tRNA molecule is going to become attached to these amino acids in the cytoplasm, and then they will be brought into the ribosome. And they have to be able to complementary base pair with the mRNA that's being read. If they don't, then they're going to get thrown out. That's how we ensure that there's a proper amino acid sequence in the protein. Okay, this just shows how the amino acid gets attached. So again, here's the enzyme that's catalyzing this reaction, aminoacyl tRNA synthetase. And according to the anticodon, phenylalanine, with the help of some energy, gets attached to the 3' end. Okay, so before I go any further, I want to make sure you understand what translation is, what a protein is, the genetic code, and what an anticodon is. So take a few minutes and fill that out for me. When you're done, you can continue.
Okay, so before you go on, just a couple more questions for you. Okay, so the first one. This is the process by which amino acids are joined together according to the sequence of bases in mRNA. Is that called transcription, translation, replication, or mRNA processing? So hopefully you answered translation. Okay, going from mRNA to protein is going to be translation, so blue there. Next question, the anticodon is found on tRNA, or the anticodon is complementary to the codon in mRNA, or the anticodon specifies the amino acid that will bind to tRNA, or yellow, the anticodon, all of the above are correct. Okay, so the anticodon, which one of those is correct? And so hopefully you answered yellow. So the anticodon is found on the tRNA molecule. Remember, it's found down there at the bottom of the T. Okay, it's 3 bases in length. It will be complementary to the codon in the mRNA because they will hydrogen bond. They'll base pair to each other. Okay, and remember that anticodon is going to be used by the aminoacyl tRNA synthase, that enzyme, long name, to grab the right amino acid and stick it onto that tRNA molecule. So yes, that anticodon does specify which amino acid the tRNA molecule will be carrying. Okay, so that's it for these 2 questions. So go to the next part, and after the next Are You With Me, we'll have some more questions.
Okay. So we've got to talk about ribosomes. What do they look like? What's their job? And of course, their job is protein synthesis. And so ribosomes are what we call them the site of translation. They are where this is happening. Ribosomes are composed of 2 different subunits, and I'll show you a picture in just a minute. Each of these subunits is going to be made up of protein and ribosomal RNA. And the 2 subunits were named beautifully for us. They're called the large and small, and that is because one is large and one is small. So that was pretty nice of whoever named that. This ribosome is going to attach to the 5' end of the mRNA transcript, and then it's going to read it from 5' to 3'. So it's going to travel along the mRNA transcript. It's going to read it 3 bases at a time. 3 bases at a time equals a codon, right? So it's going to read it codon by codon. And as it reads it, it's going to bring in the appropriate aminoacyl tRNAs, those that have the appropriate anticodon and therefore the appropriate amino acid. And as it brings them in, the amino acids are going to be covalently linked to each other, and we're going to end up building a polypeptide chain. Okay? Okay. This is what the ribosome looks like. So right here, this is a complete ribosome. So if we were to separate these 2 parts out, we have a large subunit, and we have a small subunit. And when they come together like this, the actual mRNA molecule strand is going to fit right in between, right in this groove. So between the small and large subunit, the mRNA transcript is literally held there. And then it's going to move along that RNA transcript. It's going to read it 3 bases at a time or 1 codon at a time. And there are 3 sites. And I'll show you on this one. This is larger. There's the A site, which I want you to think of as the accepting site. Okay, it's actually called the aminoacyl site, but you can think of it as the accepting site. The P site is known as the peptidyl site. This is where the growing polypeptide is going to be held. And then the E site stands for exit. Okay, this is where tRNAs, after they have already brought in their amino acid and transferred their amino acid, they're then going to leave here. They're empty. They don't have anything attached to them anymore, so they leave. So the A site is where new tRNAs are going to come in with their amino acids. And then the P site is where the growing polypeptide chain is going to be. That's the amino acid holding that growing polypeptide chain is going to be. And then the E site is where the empty tRNAs will leave. Okay. So there's 3 steps. And the steps have the same name as we named those steps in transcription. So we've got translation here. We've got 3 steps: initiation, elongation, and termination. And again, very, very similar between prokaryotes and eukaryotes, just a few key differences that we'll point out. Okay, so here is initiation. And so what do we have here in this figure? Here, this light pink, this is the mRNA transcript, right here. These little things kind of poking up off of it, those are the bases. Okay, so at the very beginning of the RNA transcript, at the 5' end, there's going to be a sequence called the leader sequence, and that's going to come before the actual gene, before the codons actually start. But the leader sequence is involved in drawing the ribosome to the mRNA transcript, okay, and lining it up in the appropriate place. What happens first is the small ribosomal subunit, shown here, is going to bind to the leader sequence, and it's going to get lined up perfectly so that the start codon, which again is always AUG, is kind of in the center of that small ribosomal subunit. And this whole process of the small ribosomal subunit binding, it's going to require these proteins that are here, called the initiation factors. Once this is set up, the first tRNA molecule, right, carrying methionine with the appropriate anticodon, which would read UAC, right, it's going to be complementary to this, will be brought in. And so that's shown right here. So this right here is the tRNA molecule. At the bottom here is the anticodon, and at the 3' end we have our first amino acid. It's methionine. In prokaryotes, methionine is actually modified. It's called formal methionine, and in eukaryotes it's just methionine. So there's one key difference that we'll actually talk about a little bit later. What happens next? Those initiation factors will come off, and that's going to allow the large ribosomal subunit to come right in and bind on top, which is shown here. Okay, and now we're all set. So the first amino acid tRNA, containing the anticodon to the start codon, and also containing methionine, our first amino acid, will be sitting right here in the peptidyl site, the P site. And the E site is now open, and the A site is open. That's initiation. Okay. So a couple of key differences for initiation if we're comparing prokaryotes and eukaryotes. Again, the first is in eukaryotes we have methionine instead of FMET, okay, formal methionine. So the methionine is just a little bit different. Also in eukaryotes, instead of a leader sequence, the start codon is actually found within a sequence towards the 5' end that indicates the site of translation. Okay, so where exactly the ribosome binds to is just a little bit different in eukaryotes. And lastly, let me actually go back here. So you can see these 3 initiation factors required for prokaryotic initiation. In eukaryotes, there's more like 10 or maybe even more than that protein factors that are involved in this initiation. So again, just more complex. After initiation comes elongation, and elongation is going to be the same in prokaryotes and eukaryotes. And so here it is in words. I'll show you a picture in just a second. So what's going to happen next is the appropriate amino acid tRNA, the one with the right codon or anticodon, will bind to the codon in the A site. Okay, and that will be a complementary base pairing and hydrogen bonding between them. So again, the codon inside the mRNA transcript and the anticodon of the amino acid tRNA are going to complementary base pair. And so that's going to bring in the next amino acid. This does require energy, and it does require other proteins. So again, we're kind of simplifying it. So let's start here at 1. This is showing initiation. Initiation actually looks like some elongation has happened as well. So what's going to happen now? We have our amino acid tRNA in our P site that's holding on to the growing polypeptide chain. Here we've got 3 amino acids covalently linked together. The A site is open. So the next amino acid tRNA with the appropriate anticodon and the appropriate amino acid is going to come in and bind in the A site, the acceptor site. Okay, that's going to require energy. It's going to be in there. The next thing that's going to happen is this growing polypeptide chain that's coming off of the amino acid tRNA in the P site is going to be transferred over here. And a covalent bond will form between this amino acid and this amino acid. And so that's shown here. So this was the amino acid originally bound to the tRNA in the A site. And here are the other 3 amino acids that came from over here. So here's the new bond that was formed. Okay, so what's going to happen next is the ribosome is going to shift. That's going to require energy. So the ribosome moves this way. And so therefore, this tRNA molecule that was in the A site, it's now in the P site. It's got the growing polypeptide chain. And the tRNA that was in the P site that had nothing now attached to it, it moves to the E site and it leaves. And this process continues and continues and continues, and this will grow and grow and grow until this is the same thing again. This is from your textbook. I wanted to make sure that I showed you some figures from your textbook as well. I like that other figure a little bit better. It's from an older textbook. So this shows initiation, right? So the small ribosomal subunit binds. Here we've got the appropriate tRNA molecule that's going to be complementary to the codon, carrying methionine. That's how we initiate. Elongation, we have another amino acid tRNA in the A site. This gets transferred over. Everybody moves down. This one leaves. This one, which was just here, now has the growing chain, and a new one comes in. And that'll continue and continue and continue until termination. And so in termination, what's going to happen is that polypeptide chain that's been growing, it is going to be released. Okay, so protein synthesis is terminated. How exactly does that happen? There is going to be a stop codon. There's 3 different kinds of stop codons. So one of them will appear in the mRNA sequence, okay, at the end of the coding region. And once the ribosome reaches that sequence, then what's going to happen is a protein called the release factor will bind to that A site, and that's going to cause the tRNA and the P site to become disassociated from the growing polypeptide chain, and that's going to cause the whole complex to come apart. So let me show you that. So here is the ribosome, right, and in this darker, almost brown maroon color, it's moving down the mRNA transcript, reading it 3 bases at a time. Here we've got our growing polypeptide chain. All these little purple dots, purple circles represent amino acids. And now we hit a stop codon. And there's 3 different kinds. It could be UAA, UAG, or UGA. When we hit that stop codon, nothing's going to bind, no tRNA molecules can bind here to the A site because there's no anticodon. And so what happens is this thing right here in turquoise, the release factor will bind to the A site. That will cause this growing polypeptide chain to be released. So it cleaves this bond right here, so that gets released. And once that is released, the whole complex now comes apart. And that's it. The growing polypeptide chain will then get folded up, and we will have a protein. Okay, so some other differences. I want to remind you that in prokaryotes, there is no nucleus. So transcription and translation are really simultaneous. In fact, we could have several ribosomes attached all at the same time to the same mRNA molecule, all reading them. That's called a polyribosome. These are pretty neat to see. There's a lot of YouTube videos too that you can take a look at where you see them. I have a cartoon for you. So in a bacterial cell, again, you have your DNA, transcription, making mRNA, and translation, all happening in the same compartment, just in the cytoplasm. And so you could get this. This light blue here is showing DNA. This is the particular region where the gene must be. Here are RNA polymerase molecules, the yellow spheres being shown here. We got 3 different RNA polymerases all attached to the same gene, all reading it and transcribing it into this pink structure here, mRNA. And then all of these light pink structures, these are all ribosomes, all attached to the same transcript before it's even being done made. And these little purple dots coming off are all the amino acids that are being put together. So we got a polyribosome, right? We've got 3 polyribosomes here. Multiple ribosomes attached and doing their job all simultaneously. Again, even before transcription is finished. So we can have transcription and translation happening at the same time. Very, very efficient. In eukaryotes, we can't have that because transcription happens in the nucleus. The pre-mRNA molecule has to be modified and then exported into the cytoplasm, and then we can have translation start. Now, there could be multiple ribosomes bound to the same mRNA transcript, but you can't have transcription and translation happening simultaneously. I wanted to also tell you about molecular chaperones, and if you had me last semester, you may have heard of these, you may not remember. Molecular chaperones, these are going to be proteins that are associated, they kind of hang out close to ribosomes, and their job is to take that growing polypeptide chain and make sure that it folds properly. So it's going to assist in folding into that final 3-dimensional shape. Without them, there could be a problem. As the amino acid chain comes off of the ribosome, it could start to fold on its own, and those interactions among the side chains could lead to improper folding of the protein. So they're there to really catalyze and help in that process. And sometimes these molecular chaperones will actually be involved in cutting a protein, maybe cutting it in 2. So there's 2 actual proteins from one mRNA transcript, or some other modification like adding a lipid or adding a carbohydrate, not a lipid, a carbohydrate. And sometimes they'll take that protein that started to fold and they'll unfold it and refold it, and there may be several folding attempts before the protein reaches its final shape. So those are called molecular chaperones. They're there to help during and after protein synthesis to make sure the protein gets
folded properly. That should have said it cut me off there. So let's go to our last Are You With Me? So here you go. You're going to want to describe the structure of a ribosome, what the steps are in translation, describe them, the key differences between translation in prokaryotes and eukaryotes, and lastly, what happens to proteins after translation is complete. This is your last Are You With Me? So when you're finished with this, remember you want to make sure that you upload it to Blackboard as either a PDF or a Word doc. And then, or as a picture, which a PDF would be great of that, or you can send me JPEGs as well. And then we just have a couple of more questions. So when you're finished with these, come back. Okay, so first question. A scientist introduces a mutation that makes the large ribosomal subunit non-functional. What will be the effect on translation? So in pink here, translation stops after the initiation codon is identified. Translation will stop during elongation. Green, the ribosome will not be able to interact with mRNA at all. Or yellow, tRNA will not be able to exit the E site of the ribosome. So take a minute and think about that. So hopefully you answered pink. So the small ribosomal subunit will be able to bind, and even that first tRNA molecule will be able to bind as well. But that's going to be about it, right? If that large ribosomal subunit doesn't come in and attach itself, we're not going to have initiation. So yes, the small ribosomal subunit can bind. It will find that initiation codon. It's going to stop there. The right tRNA molecule comes in, but that's it. So pink would be the answer. You may have thought maybe green was correct. The ribosome will not be able to interact with mRNA at all. If it was the small ribosomal subunit that was mutated, then that would be the case. But because the large ribosomal subunit doesn't bind until after, then initiation will get started. It just won't be able to be finished. Okay. Okay, next question. I like this question. Many antibiotics inhibit protein synthesis. For example, tetracycline blocks the A site on the bacterial ribosome. And a second antibiotic called chloramphenicol, it's a hard one, blocks peptidyl transfer. What specific effect would you expect each of these antibiotics to have on protein synthesis? So you may have to read that back again. Let's do the first one. Tetracycline would directly affect, okay, so tetracycline blocks the A site on the bacterial ribosome. So what would that directly affect? Would it affect tRNA from binding to the ribosome? Would it affect ribosome assembly? Or would it affect the growth of the polypeptide chain? So think about that for a minute. So hopefully you answered pink or red, whatever that color is. tRNA will not bind at all if the A site is blocked. Okay, so when you take an antibiotic like tetracycline, what you're doing is you're blocking protein synthesis in bacteria. And if bacteria can't make proteins, well, then they can't survive. Okay, so that's actually how the antibiotic kills the bacteria. Pretty cool. The second one, chloramphenicol, that would directly affect which one? So if you go back and you read about chloramphenicol, it says it blocks peptidyl transfer. Okay, so it blocks the transfer of the growing polypeptide chain. So which would it directly affect? tRNA binding to the ribosome assembly, or green, the growth of the protein chain. So hopefully you picked green. It's going to affect the growth of the protein chain. Okay, so if it blocks peptidyl transfer, then you're going to have a problem with that chain will no longer be able to grow. If it can't be transferred over to the tRNA and the A site, then it's just going to probably leave with the tRNA and the E site. Okay, so you're blocking the transfer of those peptides. So again, the same idea though. You're blocking protein synthesis, and so if bacteria can't make proteins, then they can't survive. I think that's the last question. So again, make sure you get those Are You With Me's completed. You're going to want to upload those right to Blackboard. and then your quiz is waiting for you. Okay, there are some supplementary videos in a folder on our Blackboard site, just in case you want to see things in a different way. Okay, so there are some videos there for you to look at as optional videos. Okay, so then you'll have that quiz ready for you.